Hello everyone, welcome to the new session of Morphology of the Flowering Plants, NCERT Revision. In this session, we are going to study about the fruits and seeds. Okay, so what are fruits? The fruit is a characteristic feature of the flowering plants. So we find fruits in flowering plants. Okay, only in the flowering plants. Okay, so the flower, if we draw a flower, then we are having sepals, we are having different petals, then comes the male reproductive structures, and then the female reproductive structures. So if we see the structure of female reproductive organ, then there is this is called as stigma. This stalk-like structure is called as style, and this swollen bulbous part is called as ovary. And these three structures unitedly called as pistil or carpel. Pistil or carpel. Now this ovary, this swollen ovary, after fertilization forms the fruit. This forms the fruit. And this structure is present only in flowering plant, means flower is present in the flowering plants. It is a mature ripened ovary. So here we see ovary. And when the ovary ripened, the ovary is ripened after the fertilization. Okay. So it is a mature or the ripened ovary developed after the fertilization. Okay. So it is a fruit formed without fertilization. If a fruit formed without fertilization of the ovary, it is called parthenocarpic fruit. It is called as parthenocarpic fruit. We will discuss it later, that there are some of the fruits which is formed without any fertilization, and those fruits are called as parthenocarpic fruits. But the true fruit is the one which is formed after the fertilization, and ovary, this particular part, ovary is responsible for the formation of fruit. Ripened ovary changes to fruit. Okay. Generally, the fruit consists of a wall or pericarp and seeds. So, the wall of any fruit, the wall, suppose I am drawing the mango. Here, it is drawn here, mango. So, the outermost is epicarp. The outermost is epicarp. The inner fleshy part, that is the mesocarp, and inner, this near to the seed, that is the endocarp. So the epicarp, mesocarp and endocarp unitedly called as pericarp. This is called as pericarp. This is the wall of the fruit. Okay. And the inner is seed. So pericarp and seed. The fruit generally consists of the wall of the fruit, which is called as pericarp, and the innermost seed. Okay. Now this pericarp is having three layers. The outermost epicarp, the middle one mesocarp, and the inner one is the endocarp. Okay. So sometimes the pericarp may be dry or fleshy. Sometimes the pericarp is dry and sometimes it is fleshy. When pericarp is thick and fleshy, it is differentiated into an outer epicarp, the middle mesocarp, and the inner endocarp. So we have just talked about that the pericarp can be of two different types, right? It can be fleshy, it can be dry. So here, in the case of in the case of this mango, we see that the pericarp is fleshy. The pericarp is fleshy. Okay. And in the case of coconut, the pericarp is hard. The pericarp is hard. So sometimes it is thick, it is hard, it is dry, and sometimes it is fleshy. So the pericarp is thick and fleshy, and it is differentiated into epicarp, the outer epicarp, the middle mesocarp, and the inner endocarp in the case of this mango. But here you can see the mesocarp is this hard region. Okay. Endocarp is this hard coconut shell. The mesocarp is the husk-like region which is present on the surface of the coconut. And the innermost, the white part, is the seed. So in mango and coconut fruit is a drupe. This type of fruit is called as drupe. They develop from the monocarpellary superior ovaries. So we have talked about the superior ovary, the ovary which is attached to the thalamus region, and all the floral parts like sepals, petals, and stamen originate below the ovary. Then the ovary is superior and they are one-seeded. So that type of fruit is called as drupe. This is very important. One-seeded seed, one-seeded fruit, one-seeded fruit. And it must be a superior ovary formed from the monocarpellary superior ovary. Then the fruit will be drupe, as we have seen in the case of both mango and coconut. Mango and coconut, it differs so much, okay? But they both are drupe-like structure. They are the fruit which is called as drupe. Why? Because both has been formed from the monocarpellary superior ovary and they are one-seeded fruit. Okay. In mango, the pericarp is well differentiated into an outer thin epicarp, middle fleshy edible mesocarp, and the inner stony hard endocarp. It is well differentiated, right? But in the case of coconut which is also a drupe, the mesocarp is fibrous. So here you can see the fiber-like structure. So earlier what happened when this coconut is green? When this coconut is green, it is having one cover over, one green covering. So that is the epicarp. That is the epicarp. But when this epicarp is removed, this mesocarp, you can see that is the husk-like region, brown hair, which is called as fibrous mesocarp coir. Okay, this coir region you can see. So that is the mesocarp. And the coconut and the endocarp, that is the hard brown cover. Hard brown cover. That is the endocarp. And coconut is of what we eat. What is the edible part of the coconut? That is the seed. That is the white part. That is the white part. Okay. So coconut is also called a fibrous drupe. So if drupe is the fruit, but in some some drupe fruit, the mesocarp is edible. But in some other fruit of the drupe type, the seed is edible. As we see in the case of mango, the mesocarp is the edible part. But here in the case of coconut, it is again drupe fruit. But here the seed is the edible part. False fruit or the accessory fruit. Now this is very interesting. Fruit in which other floral parts like thalamus, base of sepals and petals fuse with the pericarp is called as false fruit. So what happened? If you see the apple is like this, and this is the thalamus. This is the thalamus, and this ovary is here, the petal is here, the anther is here. stamen. Okay, sepal is here. So after the fertilization, what happened? These parts wither away. These parts wither away and detached. Now what is left? And this ovary is not up to this. It is present here. It is present here. The seeds are in the ovary. Okay. But this thalamus, so ovary is embedded in the thalamus, and this thalamus becomes swollen. This thalamus becomes swollen and it works as a fruit. It works as a fruit. So thalamus in the case of apple is the edible part. Same in the case of strawberry also. Strawberry also. This is again the thalamus. Okay. So the true fruit is the one in which ovary transforms into fruit. When the other floral parts transformed into fruit, like in the case of apple and strawberry, the thalamus transformed into fruit, it is called as false fruit or accessory fruit. Parthenocarpic fruit, in the first slide we have discussed about it, that when there is no fertilization and the ovary changes to fruit, then it is called as parthenocarpic fruit. A fruit formed without fertilization is called as parthenocarpic fruit. Parthenocarpic fruits are seedless, and if the seeds are present also, they do not give rise to a new plant. They do not give rise to a new plant. So in the case of banana, this is one of the parthenocarpic fruit. In the case of banana, we have never seen that the seeds of banana give rise to a new plant. Okay. So how the banana give rise to a new plant? So this is the giant banana, and actually the banana is a herb. It is a herb. It is also called as giant herb. It is called as giant herb. And it is having this stem, but this stem is not woody. And under the ground, there is a swollen comb-like stem part. This is the stem part underground. This is called as comb. And from this, small suckers, these are called as suckers, comes upward and forms a new plant. So in this way, like vegetative propagation, here the vegetative propagation. So this corm, if you cut and place in a soil, then again it will give rise to a new banana plant. Okay. So examples of the parthenocarpic fruit is banana. Very important. The seed. So till here we have discussed about the fruit. Now here we are going to study about the seed. The ovules after fertilization develops into seed. So where the ovules are. So if you see the ovary, then inside the ovary there is an ovule like this. This is the embryo sac. Very important. This is the embryo sac. And in the embryo sac, we are having 3 cells at this micropylar end and 3 cells at the chalazal end. Actually, this is the part we are going to study about in the sexual reproduction of the flower. Okay. And in the embryo sac, in the center, we are having 2 polar nucleus. So this is the structure of ovule. This is the structure of ovule. Now, after the fertilization, very important, after the fertilization, this ovule changes to seed. And this ovary changes to fruit. Okay. The seed is made up of seed coat and an embryo. So after the fertilization, there is a 2 times fertilization. So first fertilization, or that is called as syngamy, that in that one male gamete fuses with the egg and it forms zygote. Zygote give rise to embryo, and which later forms the plant. Okay, so first fertilization, that is called as syngamy. Now there is one more fertilization in which what happened, these two polar nuclei fuses and one more male gamete also fuses. So this is called as triple fusion. And syngamy and triple fusion unitedly called as double fertilization. Now this triple fusion, it forms endosperm, and it provides nourishment to the developing embryo, right? Okay, so seed is made up of seed coat. All the seeds are having the seed coat, and it also having the embryo. So embryo is very important. Without embryo, the new plant cannot be come out of the seed. So embryo is there inside the seed. The embryo is made up of radicle and embryonal axis and 1 or 2 cotyledons. So what we see, if we draw a dicotyledonous seed at the time of germination, here it is the radical part. So sometime what we do, we make sprouts and we put some gram soak overnight, and what happens next morning, we see a white tail like structure, a white tail or hair like structure comes out. So what is that? That is radical. And later what happened? Plumule also come out. Okay, plumule give rise to shoot system and radicle give rise to root system. Okay, and here we see 2 cotyledons. These are the 2 cotyledons. So as we see in the case of gram and pea, but only 1 cotyledon in the case of wheat and maize. Okay, next, structure of the dicotyledonous seed. The outermost covering of a seed is the seed coat. So this brown cover, this is the gram seed, which is a dicotyledonous seed example. Here we see the brown color coat, which is called as seed coat. The seed coat has 2 layers. The outer layer is called as testa and the inner layer is called as tegment. This is very, very important from the examination point of view. Sometimes these things can be asked in the examination. Okay, so the outermost layer is the testa and the innermost layer is inner layer of the seed coat is called as tegmen. The hilum is a scar. You know, there on the seed we can see a scar like structure, a mark like structure, which is called as scar, and that scar is called as hilum on the seed coat, through which the developing seeds were attached to the fruit. Okay, so in that part where I have drawn the ovule, this is the ovule structure which is present inside the ovary, and this ovule is attached to this fruit. It is attached to this fruit. So this particular part of the fruit where the ovule is attached, and this ovule later transformed into seed. So there is an attachment where it is attached to the fruit, that is called as hilum. So this hilum is that part. Okay. Above the hilum is a pore. There is an opening which is called as pore, and that pore is called as micropyle. That is called as micropyle. Within the seed coat is the embryo, and inside the seed there is an embryo. This particular part is called as embryo. An embryo, the upper part give rise to plumule, and the lower part give rise to radicle. The lower part give rise to radicle, and this unitedly called as the embryo. Okay, so within the seed coat is the embryo consisting of an embryonal axis and the two cotyledons. So here we can see two cotyledons. Two cotyledons. The cotyledons are often fleshy and full of reserve food materials. So these two cotyledons are loaded with the reserve food material, because at this particular time when this embryo is giving rise to the plumule and radical like structure, it requires lot of food material, lot of energy. Okay, so at that time the seed cannot do the process of photosynthesis, because till here the leaves are not there. So from where do they get that food material? So this cotyledon region, cotyledon part of the seed, store lots of food inside that, and that is called as reserve food material. Okay, so that reserve food material gets solubilized because lots of water has been absorbed, and when that food gets solubilized, it is available to the developing embryo. At the two ends of the embryonal axis are present the radicle and the plumule. So embryonal axis, here you can see the upper region is plumule and the lower region is the radicle. In some seeds such as castor, the endosperm formed as a result of double fertilization. The endosperm is formed at the result of the double fertilization. This also I have discussed. It is a food storing tissue and it is present in the endospermic seeds. The seeds that are having endosperm, that are loaded with the food material. Okay, and that is called as endospermic seed. Those seeds that bear endosperms are called as endospermic seeds. In plants such as bean, gram and pea, the endosperm is not present. Very, very important. Endosperm is not present in mature seed, and such seeds are called as non-endosperm. Non-endospermous. Okay, non-endospermic seeds are non-albuminous seeds. They are also called as non-albuminous seed because the albumin is the food material that has been stored in the endosperm. But when there is no endosperm, so no albumin. So no albuminous, non-albuminous seeds. It also called as non-albuminous seeds. So examples of non-endospermic seeds or the non-albuminous seeds are pea, gram, bean, mustard, groundnut, etc. Some monocot seeds are non-endospermous. These are the examples of the dicot seeds. Okay, but there are some monocot seeds also that are non-endospermous. For example, monocotyledonous seeds that are ex-albuminous or non-endospermous, we can see in the case of orchid and Sagittaria. The endosperm is consumed during the seed development and the food is stored in the cotyledons and other region. Okay, so in nymphia, now there is an example, that is nymphia, the food is stored in the nucellus or the perisperm. The food is stored in the nucellus. So what is nucellus? If you see the structure of an ovule, this is the ovule structure, this is the embryo sac. Now this part, this tissue part is called as nucellus. Nucellus. Now, in nymphia, the food is stored in this nucellus region, or perisperm. It is also called as perisperm. The seed is called as perispermous seed. So in that case, the seed is called as perispermous seeds. A layer of nutritive tissue in the seed of a certain flowering plants that are derived from the nucellus and surrounds the embryo. So here you can see this perisperm region in which the food is stored. Okay. Now, the structure of monocotyledonous seeds. So till here we have discussed about the dicotyledonous seeds. Now the monocotyledonous seeds. The monocotyledonous seeds are endospermic. Very interesting. They are having endosperm. So that is why they are called as endospermic seeds. But some in orchids and orchids and Sagittaria, we have discussed this. Orchids are non-endospermic seeds. Okay, all monocotyledonous seeds, but there are some monocotyledonous seeds are non-endospermic, such as orchids and Sagittaria, whereas generally the seeds of monocotyledonous are endospermic. Okay, so in the seeds of cereals such as maize, the seed coat is membranous. The seed coat is membranous. It's not as hard as we have seen in the case of dicot. Seed coat is having testa. Outer one is testa and the inner one is tegmen. Two layers are there. But here in the case of monocotyledonous, the seed coat is very thin, that is membranous, and generally fused with the fruit wall, and that also fused with the fruit wall. But in the case of dicot, it is not fused with the fruit wall. The endosperm is bulky and it stores a lot of food. So here you can see the endosperm structure. It is so big and the food is stored in this endosperm region. The seed coat and fruit wall, it is fused, right? And below this fruit wall, there is an aileron layer. This is aileron layer. The outer covering of the endosperm separates the embryo by the proteinaceous layer called the aileron layer. So this is the aileron layer, right? The embryo is small and situated in a groove at one end of the endosperm. So here you can see this particular part. It is having plumule region, and plumule region is having a covering over it that is called as coleoptile. And the radicle is also having a covering that is called as coleorhiza. Okay, so it consists of one large and shield-shaped cotyledon known as scutellum. So here, this particular part, this is the scutellum. This is the cotyledon of monocot. In the case of dicot, the cotyledon is laden with the food. This cotyledon here, this cotyledon in the dicot, okay, it is laden with the food material, reserve food material. But here in the case of monocot, this endosperm region is having reserve food material. The plumule and radicle are enclosed in sheaths which are called as coleoptile and coleorhiza respectively. So that also a difference if we compare with the dicot and monocot, that in the monocot we are having plumule, it is covered with the coleoptile, and radical region it is covered with the coleorhiza, but such covering is absent in the case of dicot plant, dicot seed. So here are some of the difference, dicot and monocot. Seed coat is distinct from the fruit wall, right? It is having testa, outer one is testa and the inner one is tegmen, two covering, that is fruit wall. Now seed coat is completely fused with the pericarp, okay, that is the fruit wall. There are two cotyledons in the seeds, two cotyledons in the seed, and there is a single cotyledons in the seed. Endosperm is totally absent here, endosperm is totally absent, and endosperm is present. There is no protective sheath for the plumule and radicle, but here the plumule is protected by a sheath called the coleoptile and the radicle by the coleorhiza. So this is also very important. Okay, so this is all about the fruit and seed, and one more part is left of this chapter that we will discuss in the next session. Okay, so till here we will meet again. Thank you very much.